Hello, my name is Enrique Amaya, and I'm the Professor of Tissue Regeneration and Chair of the Healing Foundation Center at the University of Manchester. Today I would like to speak to you about some work that we've been doing, which is interlinking metabolism with tissue regeneration. The primary goal of regenerative medicine research is to identify novel therapeutic approaches that will help replace injured, diseased, or aged tissues in human patients. The approach that we're taking towards this ultimate aim is to investigate the mechanisms used by some organisms to regenerate their tissues. So to begin with, we need a good experimental model organism, and mammals are particularly lousy at regenerating tissues, and that includes humans, and therefore we turn to other organisms, and the ones that we primarily like to study are amphibians, like salamanders and frogs. We do so because they regenerate remarkably well. And I'll give you an example of this. Frog tadpoles can regenerate their tails following injury, and one might think that a tail is rather uninteresting, but in fact it has many tissues that we are very interested in, like the spinal cord and major blood vessels and nerves and muscles. Following amputation, the tadpoles are able to mount a full regenerative response and regenerate all of these lost tissues within the span of a week. After a couple of weeks, you would have a hard time telling the difference between a normal tail and one that has been regenerated. So obviously we'd like to understand how these tadpoles can do that. And one of the approaches that we've taken is to try to identify all the genes that are modulated during this process. And this kind of study raised our awareness of the potential role of cellular metabolism during regeneration. So now, to be able to understand this, let's go into what metabolism does. So this is a diagram of the glycolytic pathway, which is involved in ATP production. And the primary production of ATP occurs once pyruvate goes into the mitochondria, where the jackpot of ATP production occurs. But this comes at a loss, and that loss is that you have completely combusted the glucose to carbon dioxide, and carbon dioxide is a gas which leads to carbon dioxide emissions, which is not only bad for the environment, is also bad for regeneration, as I will try to explain. Because carbon dioxide being a gas, you lose it. So by losing carbon dioxide, you're losing carbons, and therefore you're losing mass. And to regenerate tissues, you need to grow new tissues, so you need to maintain these. The way this happens is by offshoots of the glycolytic pathway, and one important one is the pentose phosphate pathway, which is involved in anabolic pathways. And these are pathways that lead to the synthesis of nucleic acids, lipids, and also amino acids. These are obviously important for tissue growth, and so now the question is, how are they regulated during regeneration? So what we found is that the first thing that you need is to import glucose, and the mechanisms by which glucose is imported are controlled by genes like leptin and insulin, and these are highly upregulated in regenerating tissues. Therefore, you get increased glucose import and also upregulation of genes involved in the transport of glucose into cells. So the end result is you get increased glucose import and glycolysis. So the other thing that you need to do is promote the anabolic pathways. And this is done through the upregulation of a critical enzyme, the glucose 6-phosphate dehydrogenase, which its expression pattern increases significantly during regeneration, and this is the gatekeeper of the pentose phosphate pathway. And by so doing, by upregulating this enzyme, you get increase of anabolic pathways. Finally, you need to limit the carbon emissions, and that is done by several processes. One is the upregulation of HIF-1 alpha, which inhibits the mitochondria, and also by growth factor stimulation, as well as reactive oxygen species production, inhibits the pyruvate kinase enzyme, thereby quieting the oxidative phosphorylation pathway. By doing so, you get decreased activity of the Krebs cycle and decreased carbon emissions. So this is the mechanisms, we believe, that are regulated during regeneration. So to conclude, I'd like to say that successful regeneration requires intricate regulation of the cellular metabolism, and that regenerative medicine approaches should take this into consideration when identifying their new therapeutic applications. And perhaps one of the reasons why some organisms regenerate better than others is because they can modulate the metabolic pathways better following injury. Ultimately, all of this research is hoping to lead to a better way of finding therapeutic approaches towards regenerating tissues in humans. Thank you for watching.